Hello, let's move on to the next section, basic pathologic changes. Although each of these stimuli may induce reactions with some distinctive characteristics in general, all inflammatory reactions have the same basic features. The basic pathologic changes of inflammation in the site of injury are alteration, exudation, and proliferation. Alteration is referred to the tissues or cells at the inflammatory sites showing degeneration and/or necrosis. This may be due to the agent that caused the initial injury, local blood circulation disturbance, the inflammatory cells and/or mediators of the inflammatory response. Morphologically, parenchymal cells show evidence of injury, for example, clotting swelling, fatty degeneration, and necrosis. Interstitial tissues also show evidence of injury, such as edema, mucoid degeneration, fibrinoid necrosis, etc. With acute viral hepatitis, hepatocyte injury takes the form of diffuse swelling. The cytoplasm looks empty and contains small clear vacuoles within the cytoplasm. Hepatic steatosis occurs in hypoxic injury and various forms of toxic or metabolic injury, and is manifested by the appearance of small or large lipid vacuoles in the cytoplasm. This picture shows coagulation necrosis, necrotic cells in the infarct. The necrotic cells show preserved outlines with loss of nuclei. Infective granuloma is caused by tubercle bacillus infection, with caseous necrosis in the central lesion. Parenchymal cells necrosis was seen in the center. At high magnification, the necrotic tissue is amorphous, granular, and pink. The details of the cells are not clear, and the structure of the tissue is completely lost. The second important pathologic change is exudation. Exudation is referred to the escape of fluids, proteins, and blood cells from the vascular system into the interstitial tissue or body cavities. When these substances accumulate in the interstitial tissue, we call it edema. When accumulate in body cavities, we call it hydrops. The process of exudation includes changes of hemodynamics, increased vascular permeability, and exudation of fluids and leukocytes. Initially, there is transient constriction of arterioles. This change only lasts for a few seconds. Then, blood vessels dilate, capillary base expands, opening of new capillary base increased blood flow. This vascular expansion is the cause of redness and warmth, two cardinal signs of inflammation. These processes are induced by mediators such as histamine, tannin, or axon reflection. After a transient vasoconstriction, blood vessels dilate and blood flow increases. The microvasculature becomes more permeable, and protein-rich fluid moves into the extracellular tissues. This causes the red cells in the flowing blood to become more concentrated, thereby increasing blood viscosity and slowing the circulation. Numerous small blood vessels are packed with red blood cells. We call it stasis. As stasis develops, leukocytes, principally neutrophils, begin to accumulate along the vascular endothelial surface, a process called margination. This is the first step in the journey of leukocytes through the vascular wall into the interstitial tissue. The picture shows vascular and cellular reactions of acute inflammation. Compared with normal, the major local manifestation of acute inflammation include vascular dilation and increased blood flow, causing erythema and warmth, leakage of plasma fluids and proteins, causing edema, and leukocytes, meaning neutrophils, emigration and accumulation in the site of injury. In the early stage of inflammation, vascular dilation and increased volume of blood flow lead to a rise in intravascular hydrostatic pressure, and then fluid leak from capillaries into the tissues. This fluid, called a transudate, is essentially an ultrafiltrate of blood plasma and contains little protein. However, transudation is soon replaced by exudation. Increased vascular permeability allows plasma proteins, leukocytes, and even red blood cells to enter sites of infection or tissue damage. This fluid, rich in protein and cells, is called an exudate. Increased vascular permeability is an important cause of fluid and protein leaking from blood vessels. During inflammation, the following mechanisms can cause increased vascular permeability. When stimulated by inflammatory mediators such as histamine and bradykinin, endothelial cells rapidly contract, resulting in gaps between endothelial cells and increased vascular permeability. This process is short-lived and usually occurs in post-capillary venules. A slower and more pronounced retraction of endothelial cells resulting from changes in the cytoskeleton may be induced by cytokines, such as tumor necrosis factor and IL-1. This reaction may take 4 to 6 hours to develop after the initial trigger and persist for 24 hours or more. Direct endothelial injury is usually seen after severe injury, such as burns, toxins, and some chemicals. It results in endothelial cell necrosis and detachment. Leukocyte-mediated endothelial injury may occur as a consequence of leukocyte accumulation along the vessel wall. Activated leukocytes release mainly toxic mediators that cause endothelial cell injury and detachment. Increased transcytosis is another mechanism of increased vascular permeability. In addition, new capillaries have high permeability. tissue repair involves new blood vessel formation. These new blood vessels is immature and may have some leakage from intercellular junction. The outcomes of the increased vessel permeability include fluid exudate and leukocyte exudate. When fluid accumulates in interstitium, we call it edema. When accumulated in body cavity, we call it hydrops. This picture shows inflammatory edema, fluid accumulation in interstitial spaces. This picture shows peritoneal hydrops. The liquid is cloudy and white. This is exudate. In clinical practice, exudate needs to be distinguished from transudate. The table shows the differences between them. First of all, they are produced under different conditions. Whereas exudates are typical in inflammation, transudates accumulate in non-inflammatory conditions. The transudate has a clear appearance, no fibrin, few proteins, and cells. The gravity is lower than 1.018, and the rewater test was negative. The exudate has a cloudy appearance, contains fibrin, a lot of proteins and cells. The gravity is more than 1.018, and the rewater test was positive. The protein-rich fluid exudate in inflamed tissue contains antibodies, complement, and lectin, which coat bacteria before phagocytosis, and also contain fibrinogen, which is converted to cross-linked fibrin. There are some beneficial effects of the fluid exudation. The exudate can dilute local toxins and reduce the injury to tissue, bring in the nutrition substance for leukocytes, and carry off the metabolic products in inflammatory foci. Furthermore, the exudate that contains antibodies and complements can kill the passengers. In inflammation response, fibrinogen is converted to fibrin. Fibrin is important in inflammation. Fibrin can not only limit the spread of pathogens, but also provide surface to facilitate phagocytosis. In later stage of inflammation, fibrin provides scaffold for tissue repair to promote fibroblasts producing collagen. Besides, exudate may produce humoral immunity. However, too much exudate can cause compression, obstruction, and adhesion. For example, the laryngeal edema can cause obstruction of throat and suffocation. Leukocyte exudation is the most important feature of the inflammatory reaction. It involves leukocyte margination, adhesion, immigration, phagocytosis, intercellular degradation of ingested particles, and extracellular release of leukocyte products. Neutrophils, eosinophils, basophils, and monocytes, and lymphocytes all use the same pathway. There are several steps involved during the leukocyte exudation, include margination, rolling, adhesion, transmigration, and migrate toward the site of infection or injury. The first step, margination and rolling. As blood flow slows, leukocytes enter the periphery of the blood stream, coming to lie close to the endothelium. Endothelial cells express selectins such as P-selectin and E-selectin, and interact with receptors on the leukocytes, binding the leukocytes to the endothelium. Selectins bind to the receptors and then separate from the receptors and mediate leukocytic rolling on the surface of endothelium. The second step is adhesion. High affinity states of integrin expressed on the surface of leukocytes combine to the integrin ligand and mediate firm adhesion of leukocytes to the endothelium. Then, PECAM1 expressed on the surface of these two types of cells mediates leukocyte migration through the blood vessel wall. Following extravasation, leukocytes migrate toward the site of infection or injury along a chemical gradient by a process called chemotaxis. The active process involved amoeboid cell movement. Okay, look at this picture. In the center of the picture, there is a blood vessel. In this area, you can see some neutrophils close to the endothelium. This is margination. And here, these cells are going through the wall. That's transmigration. In addition, there are many neutrophils infiltrate in the extravascular tissues. Immigration is refers to the process by which motile leukocytes migrate out of the blood vessels. Although all leukocytes are more or less motile, the most active are the neutrophils and the monocytes. The lymphocytes are more sluggish. Leukocyte immigration is an active, energy-dependent process. Red blood cells out of blood vessels called diapedesis is believed to be passive. Diapedesis is due to the loss of red blood cells through the points of vessel rupture. Leukocytes are out of blood vessels that are known as inflammatory cells. Inflammatory cells include granular leukocytes and non-granular leukocytes. Granular leukocytes include neutrophils, basophils, and eosinophils. Non-granular leukocytes include monocytes and lymphocytes. Neutrophils are commonly seen in early stages of acute inflammation and purulent inflammation. Neutrophils granules contain lysosomal digestive enzymes and reactive oxygen intermediates. Look at the picture. The four cells are neutrophils. In the cytoplasm, there are many small pink granules, and the nucleus is lobulated or rod-shaped. The four cells are eosinophils. The cells are round, have lobulated or rod-shaped nucleus. The cytoplasm contains large orange-red granules. Eosinophils are commonly seen in hypersensitivity reaction and human parasite infection. Basophils participate in acute and persisting inflammation by release of histamine and oxidative products from their granules, commonly seen in hypersensitivity reaction. Mast cells may produce fibrogenic cytokines. Leukocytes and plasma cells are commonly seen in virus infection and chronic inflammation. The two cells are lymphocytes. They are small, have a dark blue nucleus and little cytoplasm. The two cells are plasma cells. Plasma cells have a round, eccentric nucleus with coarse clumps of chromatin. It looks like a soccer ball. Macrophages, the name means big eaters. Tissue macrophages are derived from blood monocytes that immigrate from blood vessels under influence of chemoattractants. Macrophages commonly seen in the later stages of acute inflammation, chronic inflammation, and non-purulent inflammation, and viral or protozoal or fungal infections. In some situations, macrophages assume distinctive forms. Look at the picture. All the cells are derived from macrophages. Dust cells containing phagocytosed-like dust are found in the alveoli of coal miners. The macrophages that swallow typhoid bacilli, red blood cells, lymphocytes, or cell debris are called typhoid cells. Foam cells containing phagocytosed lipid can be found in atherosclerotic plaques. Epithelioid cells are characteristic regions of tuberculosis. They also contain lipid. Langerhans-type germ cells containing more than 20 nuclei are also found in tuberculosis. Heart failure cells are found in the sputum of patient suffered from left heart failure. These cells contain granules of hemosiderin derived from broken down red blood cells in the alveoli. Foreign body germ cells can be seen in the chronic inflammation caused by foreign bodies. The cell is very big with multiple nuclei, and the nuclei are arranged disorder. So all of the cells are essentially macrophages. The functions of leukocytes include phagocytosis, immunological function, and leukocyte-induced tissue injury. Phagocytosis is the process that leukocytes phagocytoses pathogens, tissue debris, and foreign bodies. Both neutrophils and macrophages have strong phagocytosis, and eosinophils have weak phagocytosis. Leukocytes immigrate to the site of inflamed, engulf, and phagocytose bacteria and other organisms, then kill or degrade the pathogen and tissue debris. It's a multi-stage process involving. Phagocytosis consists of three distinct but interrelated steps. First, recognition and attachment of the particle to the ingesting leukocyte. Second, engulfment, with subsequent formation of the phagocytic vacuole. Then, killing and degradation of the ingested material through the release of reactive oxygen intermediates, or by the action of enzymes stored in the leukocyte granules. The third basic pathologic change is proliferation. In the presence of inflammatory factors, parenchyma cells and stroma cells can proliferate. Proliferation of endothelial cells, macrophages, and fibroblasts is a major component of the inflammation. Now, let's make a summary for this section. In this section, we have talked about the basic pathologic changes. The basic pathologic changes include alteration, exudation, and proliferation. Exudation is the most important feature to the inflammatory reaction. We have talked about the mechanisms and the process of exudation. That's all for the section.